So if you are writing exam, make sure you stay to the end and make sure you subscribe to the channel. This NECO Biology 2026 Practicals. Stay tuned. Make sure you watch to the end to get every details about what we're about to do here. So the question say, identify specimen A, B, C, D, and E. So specimen A is the pelvic girdle of a rabbit. Pelvic girdle of a rabbit. That's specimen A. You will see it either by drawing or physically of a rabbit. Specimen B is a cervical vertebrate. Cervical vertebrate of rabbits. Take note. There's a difference between the two. Specimen C, we have the cordia, vertebra of a rabbit. That's specimen C. Then specimen D, we have the transverse section. Transverse XE, transverse section of pawpaw. Then specimen E, we have the transverse section. Transverse section of a guava fruit. So this is specimen A, B, and C. Then Roman figure 2, you still identify the location of each of A, B, and C in the body of the animal that possesses them, which is the rabbit. Where can we find the pelvic girdle? It can be found around the waist. Or the pelvic region. Here you see the pelvic girdle. That specimen B is found around the neck region. And specimen C is found around the tail region. That is where you see specimen C. So let's go to Roman figure 3. It says, name the 3 bones that make up A. We have the pubis. P-U-B-I-S. Please take note of your spellings tomorrow, because those are the errors they mark in biology. We have the pubis, we have the ischium. Then they also have the ilium. These are the things they look out for in biology. So let's go to Roman figure 4. It says state 2 structural differences between B and C. Structural difference, what do you see between them? So let me put them down here because of space. For B, I will write B and C and separate them. You have a neural spine. Neural spine is reduced. For the C, neural spine absent. Neural spine is absent. That's one of the structural differences between both of them. Then the second one, we have the transverse process is wing-like. That is for specimen B. While for specimen C, transverse process is absent. So that is the Roman figure 4. Let's go to Roman figure 5 of the question. Yes, here we are. This is Roman figure 5 of the question. So let me bold it in so you can see very well. Then it's a state 2 function of B to the organism that possesses it. 2 function of B to the organism that possesses it. It allows passage of vertebral artery. It allow passage of vertebral artery and vein. You can also include the vein, artery and vein. Then number 2, it allows movement of the head. It allows movement of the head. Allow movement of the head. So take note. Then question B of it, we are asked to state the types of placentation observed in D and E. We have for the D, we have the parental placentation, Then in the E, we have the axial placentation. So that goes for 2 marks. Then B, Roman figure 2, we are asked to state one reason for each of specimen D and E. Okay, for specimen D, the ovules developed directly on the inner wall. Directly on the inner wall. Take notes. Inner walls of the ovary. For E, let me put it here. The ovules are attached to the central axis, to the central axis. So that is that. Then question C says you draw a label diagram of 8 to 10 cm of specimen E. So this is the diagram. You can look it out. 8 to 10 cm of specimen E. This is the diagram. That is question 1C. So take your time and label directly. If you are labeling, you put a single arrow and ensure your lines are single. Don't do double lines in this manner. No, don't do double lines in this manner. Ensure you have a single line. Then when you label, don't put a straight line. Don't label in this form, No, ensure you have a straight line in this manner. I point to the part you want to label. So that is the solution to that question 1C. And you must make sure you measure from here to here. between 8. Maximum length is 10 cm. Here also, maximum length is 10 cm. You must measure with your ruler to ensure you fall between 8 to 10 cm. And make sure your labeling is as neat as well as you are seeing it. So let's go to question 2. Let's go to question 2. So look at what we have here. It says identify specimen F, G, H, I, and G. So for specimen F, we have the queer feather. For specimen G, you have the agama lizard. Then specimen H, you have the rat or rabbit. Let's use a rabbit, or you can also be a rat. The specimens I, you have the quadrant. And specimen J, you have the sweeping nets. That is specimen G. So follow them through. Then remember 2, it states three importance of F to the organism that possess it. 1, it aids, which is the queer feather, and it aids flight. Then 2, protect the bird from sunlight. It protects the bird from sunlight. Number 3, it aids in temperature regulation. Then remember 3, we are asked to mention 3 adaptive features of G to its environment. 1 is number 1, we have the claw digit. We have the claw digits for climbing. And the claw digit for climbing. Then 2, we have the dry scaly skin. SA, dry scaly skin for protection. Then remember 3, and we have sharp homodont. You have sharp, which is the teeth now, homodont. Sharp homodont teeth for showing. You use that for showing. That is Roman figure 3. Let's move to Roman figure 4. It says, state the class each of G and H belong. G is reptilia. It's reptilia. Is reptilia, and H is mammalian. So that is for G, and let's go to H. Let's go to Roman figure 5. We have Roman figure 5. Just give two natural differences between G and H. One, presence of scale. I'll write here presence of scale. Then this other one, we have a scale is absent. Scale is absent. Then the second, we have absence of body hair. Absence of body hair. In the second, we have presence of body hair. Presence of body hair. That's the difference between both of them. Then let's go to Roman figure 4. Roman figure 4. It says state three common features in G and H that serve as evidence in convergence revolution. We have pretensity, then 2, possession of 4 and hinge. Possession of 4 and hinge limbs. Then number 3, sensory organs like ears. Sensory organs like ear. Sensory organs like ear is what they have. So that's Roman figure 4. So let's go to Roman figure I. Let's go to I. So what we have there? It's a mention on function of I and J. Function of I and J. I to estimate population of small plants. That's what I to estimate. Population of small plants. Then for catching, flying or jumping insects. For catching, flying insects. That is it, for catching, flying insects. Now, Roman figure 2 of that, you say, describe in 3 steps how I is used to perform the function listed in B Roman figure 1. So look at the function there. How do we use I to perform that? So let's see. That's Roman figure 2. The place, I'm going to say place quadrant randomly. Place quadrant randomly on a specific area. That is it. Then 2, collect data either by counting, collect data by counting. Then 3, estimate the total population. Estimate the total population. Estimate the total population. Okay? So by using a mathematical formula, which is total study area over quadrant area times the average number per quadrant. That is the formula. of estimating the total area. Let's go to the next question, which is Roman figure. So identify key L, N, and the O. For K is housefly. Then A is pride of Barbados. Pride of Barbados. Pride of Barbados. Then M, cashew nuts. Cashew nut. Then N is millipede. Millipede. Then O is crab. Then P is soya grain. That's that. So let's go to, now we go to say give 3 adaptive features of K. One, it has a compound I. Compound I for wide, for vision. Let's not put details for vision. Compound I for vision. The 2, you have membranous wing. Membranous wings. You know, for flights. Let me complete it for flights. Then next, you see the pair of jointed legs. Pair of jointed legs. Pair of jointed legs. Then Roman figure 3, say mention one economic importance of K. One economic importance of K is the process of decay. It aids the process of decay. It aids the process of decay. Then question B part of it is name the class each to which K, N, and O belong. K is in center. Then N is Myriapoda. And O is crustacean. O is crustacean. So let's go to Roman figure 2 of B. Go to Roman figure 2 of B. It said, state the habit of K, N, and O. For K is terrestrial habitat. Terrestrial habitat. N is also terrestrial habitat. Then O is aquatic habitat. It's aquatic habitat. Then classify L, M, and N into their fruit type. So classify M, N, and N into their fruit type. Number 1 is L is legume or pod. Then M is nut, and P is Caiosis. Yes, that is for P. Let's go to C, Roman figure 2. It says state the mode of dispersion for L and M. So before we go to question C, Roman figure 2, make sure you share, like, and subscribe to the channel to get other exciting videos just as that will help you in your exams. Don't forget to subscribe. So you should go to state the mode of dispersion of each of L and M. For the L, it's explosive mechanism. Then the M is animals, help in that dispersion process. We have animals. Then question number 3, it says state 2 observable differences between N and O. Observable differences between N and O. 1, you have breathe through this breathes through these barnacles. Okay, this arrow A. We have CLES. So this one breathe through to the gigs. So let's take number 2 here because of space. So we have number 2, length ranges, it leg range from 40 and above. 40 and above. Second case, legs exactly 10 legs. Exactly 10 legs. With two serving as claws. With 2 serving as claws. So that is the solution to the question. If you like what you are watching, subscribe, like, and expect video from this channel. So stay with us as we make sure you draw correctly. Spell correctly is very important, and make sure you do the measurement of the drawing to obtain the full marks required in the exam. So that's what you should do tomorrow, which is this morning, and make sure your pencil is thick and sharp. So see you in our next video. Bye for now.